partly why many of you are here. So this is a fairly new direction for my lab. It was kind of kicked off by Ann Royer, who's now a postdoc at Willamette University. My current student, Sam Perez, is working on it now, and we're collaborating with Tom Mueller at Boston. So I think the study of phenotypic evolution is properly focused primarily on adaptive traits, and I say properly because, well, I've spent my whole career doing that, and if I didn't think it was, I could do it or something. So also, adaptation is the fundamental process, right? All life on Earth depends on adaptation. However, we know that cases of loss of trait function are common, and it's usually due to some kind of ecological change. The poster child, these are some examples from a Lottie et al. Paper from a Nesson working group. Poster child, cavefish, they become troglodytes and they lose eyes and pigment. A floral one that I like is Kim Steiner's work on some South African orchids, where this species has these oil glands, and that's the reward for the bumblebee pollinators. There's a derived species that's pollinated by beetles, and they either have vestigial oil glands or no oil gland at all. So when traits lose function, we think selection then it is relaxed. But in a lot of the best known examples, like cavefish and stickleback armor, it seems that the loss is pretty rapid and controversial, but may be driven by selection. So the example I'm going to talk about today, we think selection is likely weak. We don't have direct evidence for that, but that's the scenario. And partly the evidence for that is loss is incomplete. And with weak selection, adaptation is more likely to be constrained by things like drift, pleiotropy, and epistasis. And I've argued that despite a lot of interest in genetic constraints on adaptive evolution, I don't think we really understand it too well, and this could be one reason. We know that strong selection can overcome all kinds of what should be pretty strong constraints. And so I think weak selection might be an ideal system to understand the interplay among these fundamental evolutionary processes in phenotypic evolution. So the trait I'm talking about, there's the botanical word, so it's a plant crowd. So, but tetradinomy just means 4 plus 2, and so this is wild radish that I spent most of my career on, where it means that there are 4 long stamens. Those are the anthers there that make the pollen, in case there's a few non-pollens. And then there's 2 short stamens. There's the anther of one of them there. And so we've done quite a bit of work in this outcrossing wild radish species, and we don't know why they have 4 long and 2 short stamens very well. In fact, so mustard brassicaceae, almost 4,000 species. This is a diagnostic trait, so we really don't understand much about this trait in outcrossers. But I'm not going to talk about that today. Today, I want to focus on, of course, one of the most common evolutionary transitions in the angiosperms is the transition to selfing, and so this has happened in Brassicaceae too, including this little-known plant, Rhabdopsis thaliana, which has high rates of selfing, around 98% typically, and recently derived from an outcrossing ancestors. So I really like this false color SEM that we stole off the internet, because it shows what's happened here. So here, so here are the long stamen anthers, the yellow things are the pollen, and you can see these long stamen anthers are perfectly positioned to deposit that self-pollen autonomously on the stigma there. That's the stigma that everything with its papillae, it's not hairs. And this happens autonomously. It often even happens in the bud before the flower opens. Here's the one of the short stamens, and there's its anther placing its pollen on the side of the style. And that doesn't work too well, and Anne has shown through experimental stamen removal experiments that the short stamens do not contribute significantly to self-seed set. So it seems to have lost most or all of its function, at least for selfing, and so what should happen is those short stamens should be lost, right? And that's what Anne found, and in fact there's a latitudinal climb. So here we have latitude across the European range, and I think there are 30 populations here. We have more now, the same pattern holds, where at low latitude there is stamen loss somewhere. So this is the Mediterranean, the southern end of the native range of Rhabdopsis. So on average around 1 short stamen left. Nobody's down there at 0. Okay, so there's no population with complete stamen loss. And then up here in Scandinavia, stamen loss is rare. They pretty much have both of their short stamens in all their flowers. So some fun facts for stamen loss. See how charismatic this plant is? This is actually an old, drawing from England. It's a little bit actually there. But so the loss of individual short stamens is complete. So we almost never see like a partial short stamen. They're either there, the individual stamens, or they're not. Long stamens are very rarely lost. Okay? So it's a complete loss of the stamen when it happens, and it's almost always the short stamen. But also, so I said loss is never complete within populations. You saw that in that graph. But it's really cool that it's never complete within individual plants, at least that we've found so far. So here those things are flowers there. I don't know if you can see them, but they're tiny little flowers. So this one may have 0 short stamens. This one may have 2. These up here may have 1. So the flower is a serially repeated structure within each flower because the loss of the stamens is complete. There's only 3 phenotypes, 0, 1, or 2 short stamens. But then if you look over the whole flower, since the whole plant, since there's variation among flowers, a mean stamen number at the whole plant, the individual plant level is a quantitative trait. So it's pretty cool in that sense. And what Sam Perez has found is that latitudinal climb that Anne found is paralleled by an altitudinal climb. These are the populations that Alicia Montesinos and Steve Tonser and others have studied in Spain. And so what Sam finds, again, parallel to the latitudinal climb is at low altitude, below 500 meters, the populations tend to have about 1 short stamen. And then it rises to an asymptote at about 1,000 meters. You plateau not at 2, but about 1.8 on average. There's one population so far that seems to have almost no short stamen loss. Interesting, intermediate altitude. So, some hypotheses for these climbs. We don't have much data, so I'm just going to mention them briefly, and we can talk about it afterwards if anybody's interested. One, of course, is that there's direct selection on stamen number that varies with latitude and altitude. Another, that there's constraint due to drift. And I can talk about why that should be altitudinal and latitudinal later. What I'm going to talk about a little more today is constraints due to pleiotropy, because we know that there's lots of traits that show these parallel altitudinal and latitudinal climbs. And so if there's pleiotropy with one of those traits in stamen loss, that could be explaining these climbs. And so what we've done so far is mostly to look at genetic mechanisms as a first step at getting at this. So again, in Anne's work, here's her slide. Fortuitously, this is the Italian, sorry, it's high latitude, that's not Italy. The Swedish plant population that Doug Shemsky and Junagren have used in their studies. There's the Italian plant, so the Swedish plant has almost no stamen loss. The Italian plant has lost on average about 1. Shemsky lab crossed those, created this great set of recombinant inbred lines. It really is nice to work in inbred plants. I've never done it before, but that's a big reason. They genotyped them and didn't need to do any of that. She just needed to score stamen loss to find the genomic regions affecting loss. I mean, she did score kind of a lot of flowers. That's cool. But it's a pretty simple trait to score, so that's nice. So this is a standard QTL plot where what we see along the x-axis, these are the 3 chromosomes where Anne found a QTL. There are only 5 in Arabidopsis. And then the y-axis is the log odd score, giving the statistical support for a QTL that is a gene region affecting stamen loss. And so because of lots of flowers and lots of recombinant inbred lines that can be replicated with replicate individuals within each line, and had really good power. So here's a weak one on chromosome 1, and explain less than 3% of the variance among the rills, and the difference between the homozygotes, the 2 alternate homozygotes, was about 0.1 short stamen, so it's a small effect. I guess maybe some people call that, but anyway, I won't get into that. An intermediate one on chromosome 3, and then a whopping big one on chromosome 5 with the 2 homozygotes. Well, it's still there. The 2 homozygotes explain a third of a stamen. So I talked about pleiotropy. The Shemsky lab has been working on these rills a lot. Junagren and Doug and Chris Oakley and some others did reciprocal transplant of these rills in Sweden and Italy, and they found fitness QTLs for fruit production over 3 years, and one of those co-localizes with this one on chromosome 5, and Emily Dittmar, a student of Doug's, has looked at flowering time. There's a co-localized QTL for flowering time in these lines right there too, and also FLC, one of the major well-known flowering time loci in Rhabdopsis is there too. So and of course flowering time and fitness, we're talking about fitness differences across the latitudinal gradient. So these are candidates for pleiotropy explaining the decline. And we found both those, plus a freezing tolerance locus and co-localized with a QTL on chromosome 1. Then we searched for other parents of real sets available. There are lots of them in Arabidopsis, pretty cool. That differed in stamen number, and we found Sue and Cass, which is Tom Junger and John McKay and others have developed this set to look at drought tolerance, but fortuitously, there's stamen loss among the parents. Well, one parent has stamen loss, the other does not. And we found 4 QTL, this is Sam's work, 4 QTL there, and I can overlay this, and 2 of them overlap broadly, the one on chromosome 1 and the giant one on chromosome 5, and it's a giant one in the Sue Cass real set as well, explaining lots of variants. So there's some repeatability in these 2 independent crosses. And then the next thing, I think, so the thing that we didn't have to do anything for, that the Junger, McKay, and Shemsky labs have sequenced all 4 of the parents for these 2 crosses. Okay, so the parental sequences are known, and so what Sam did is focus first, this is literally over the weekend, on the big QTL on chromosome 5 and search for parallel substitutions in the overlap region. That is, the substitutions where both the 2 independent parents that had stamen loss had the same nucleotide, and there was a change, a SNP, compared to the 2 parental populations that did not have stamen loss. And he found 35 shared SNPs in the overlapping range of the 95% confidence, incredible interval, this QTL. 35 shared SNPs between those 2 parents with loss. 25 of them are clustered in this pretty small 1.2 kb region in 29, so naturally it must be a good transcription factor. And of course, that's a pretty good candidate locus for pleiotropic effects. I hope I've convinced you that this might be a pretty good system to study trait loss and interplay among different evolutionary forces. Some of the advantages, just the ones that I've talked about today, we've got these clines, which are pretty cool, and stamen loss is not complete, so there's lots of intraspecific variation for us to work with, which is not true in some of those trait loss systems. We've got QTL, 2 of the QTL are repeated across populations, one's really big. There's pretty preliminary evidence for pleiotropy. There's suggested evidence for pleiotropy still at the QTL level, right? There are a lot of genes in those QTL intervals, but we do have a pretty reasonable candidate gene now. These things often fail to work out, but it's worth following up on for sure. And so with that, thank the people in Doug's lab, John's a postdoc in Tom Junger's lab, Cindy, my technician and an undergraduate, and Rob Last is a collaborator, a colleague of mine at Michigan State, and I'd be happy to take questions. Tom.
I think there are mid-29 knockouts today.
As I say, this was over the weekend. Sam sent me this for Sunday. So, I mean, there's salt knockouts for almost everything, but maybe those are better. So, yes, that's a good thing. Can you send Sam an email and tell him? Speaking of Sam.
So, Arabidopsis is completely selfing. Is there a decline in outcrossing?
That's part of the direct selection hypothesis. We kind of expect it to go the other way. And Dina Grossenbecker has a little bit of preliminary evidence that selfing rates, I can't remember the tax, it might be across multiple taxa, are higher at high latitudes and altitudes. And we might expect that, the selfing syndrome, rapid life cycles, which you need at high latitude and altitude. Of course, this goes the wrong way, but retention, and there may be fewer pollinators. Very little is known about pollination in Thalliana, but when people look, it's pretty rare. Those little flowers are really not very attractive. But it's a hypothesis that was in my pre-proposal.
One really quick question.
Or are you quick?
I don't know if I'm quick. So you set this up as talking about weak selection, but when you talked about the other traits that these could be pleiotrophic with, they're important.
Right, so weak selection directly on the stamen loss trait, and so that's a case where the pleiotropy can really, I think, take over, and that's what I meant. So I don't think there's strong selection on these short stamens.